All right, so now we will talk about dysbiosis, and this is when the community structure changes in a way that removes one of its functions. So what does that mean? What is community structure, and what are the beneficial functions? Well, the beneficial functions are the things I might have mentioned, like protecting us from pathogens, and there are others. What is community structure? Well, it's the pattern of abundance of every species. So community structure is the overall term we use for describing which species are abundant, how abundant are they. So if you've got, I don't know, bacillus that is extremely abundant, and you've got streptomyces that is half as abundant, those measurements together is part of the community structure. And so if you list how abundant all the different bacteria are, how abundant all the different fungi are, that is a snapshot of the community structure. And as any one species gets more or less abundant, that is a change to the community structure. Community structure is changing always a little bit. That's one of the things that makes it challenging to study the human microbiota. The community structures are always changing, but they generally change within certain bounds and certain rates. And often one species or strain will disappear and be replaced with another that serves the same function. And so we don't notice that that change is taking place. So some examples of dysbiosis are, again, dysbiosis is when the community structure changes and that leads to a loss of function. So some examples would be if someone is infected by Clostridioides difficile, or C. diff, that typically happened in the past when antibiotics would disrupt the gut bacterial community. So broad-spectrum antibiotics like clindamycin would wipe out most of the bacteria in the gut, and C. diff would be able to grow in that condition. So what that tells us is that under normal conditions, C. diff couldn't grow because it was being either directly attacked or outcompeted by the typical members of the microbiota. So the microbiota was protecting us from it. And when we screwed up the community structure, we lose that protection. The same thing, or not the same thing, but something similar can happen when the vaginal microbiota is disrupted, and that can lead to, or that is bacterial vaginosis. So bacterial vaginosis is not very well understood, but it's when we can detect significant disruption to the normal microbiota of the vaginal mucosa. Well, that disruption correlates to higher susceptibility to HIV and other sexually transmitted infections. So, bacterial vaginosis, again, is the condition sometimes when sometimes leads to itching or a discharge or a fishy odor. Sometimes it doesn't have any of those signs at all, or symptoms at all. But either way, it leads to less protection against other diseases. And so that's a situation where just changing the bacterial structure, or bacterial community structure, can have a real effect on our health. This is an experimental demonstration of this, where the researchers were studying vancomycin-resistant enterococcus, so VRE, vancomycin-resistant enterococcus. You can think back to Lecture 8. And those could colonize the guts of mice. So what they were looking at was what will happen if you give antibiotics to mice and then wait 2 weeks or 4 weeks and let the bacteria regrow after wiping them out with antibiotics. What will happen? So what they did was, first, they took some untreated regular mice, and fed these bacteria. They fed them vancomycin-resistant Enterococcus. And then they measured how much took up residence in the gut. Did they grow? Were they detectable? And so in both the small intestine and the cecum, and the cecum is sort of like the large intestine of a mouse. They don't have the same gut we have. But this ND means not detected. And so this level here is the minimum they could detect. So they graph it as here, minimum, not detected. That is, when normal mice ate vancomycin-resistant Enterococci, they did not colonize the intestine. They did not colonize the cecum. We know the number is somewhere below the limit of detection. So then, some similar mice were given antibiotics and then immediately treated with, or immediately given vancomycin-resistant Enterococci. So the antibiotics would disrupt the normal gut bacterial community. And here's what we saw. When right after antibiotic treatment, the mice had no defense against the vancomycin-resistant Enterococci. They went from not detected, like 10 to the first power here, all the way up to 10 to the 6th or 10 to the 7th. This is probably about 3 million cells. So from less than 10 to 3 million, and from less than 100 to 300 million in the cecum. So, and what these little stars indicate is that there is a detected significant difference between these numbers. So then the question, that is what they expected. Untreated mice have a defense. The bacterial community protects them against VRE. If you give the mice antibiotics, that protection is gone because the bacteria that would compete are gone, and so VRE can colonize. But then the question is, what if you antibiotic treat the mice and give them 2 weeks to recover? Give their gut communities 2 weeks to grow back. That's more than enough time for the bacteria to grow back. And so these mice, after 2 weeks, would have a full complement of gut bacteria. Their microbiota recolonize their guts. So then the question is, what will we see? What will we see? Would they get back that protection so that the VRE would be undetectable? Or do they have no protection, just like they had right after getting antibiotics? And essentially they had less protection, much less protection than healthy mice had after 2 weeks or after 4 weeks. What this is showing us is that the antibiotics on a long-term scale decrease the protection the mice got from their microbiota. So the new bacteria that grew back in the gut after the antibiotics were taken away were not the same as the bacteria that had been there. So this is an example of dysbiosis. Antibiotics permanently changed the community structure. And we could detect that through a loss in function, a loss in protective function from the community. So there's that. Yeah. So some other things we think dysbiosis could correlate to. This is not a complete list, but all of these different things have been correlated to changes in the bacterial community structure in the gut. Things that are more obvious would be things like constipation and susceptibility to gut pathogens. But also cancers, also heart disease, and more recently, I don't have access to these papers yet, but it looks like there are connections with things like neurological disorders. We'll get to that later. As I mentioned in class, fecal microbiome or microbiota transplants are the only real treatment we have for changing the microbiota or reversing dysbiosis. This really is the only thing that can directly address dysbiosis. And what this is is putting a new bacterial community in the gut and hoping it stays there. So it's not reversing the damage to the original community. It's trying to replace it with a different community that is more functional. So I think it's hard for me to describe the ecology of bacterial communities without using analogies. So I think of forests and prairies. So I'm from Illinois, where the prairie is what I understand the best, but I think in most places a forest is understood better. So if the trees in a forest and the smaller plants in a forest and the birds and the what else is in a forest? Squirrels and insects and spiders and all that different stuff, everything in the forest. If that is the bacterial community in the gut, then a fecal microbiome transplant is digging up a different forest and planting it in this forest. So using this analogy, dysbiosis would be a situation where something happened to some of the trees and the animals aren't getting the food they need. And so the forest has clear patches it didn't have before, and you're not hearing the birds singing, and you can't harvest wood or something. I don't know. That's dysbiosis. Something has changed in the forest and now it's not doing what it was supposed to do. So fecal microbiota transplants are kind of crazy and they are dangerous, but in some cases they're the best thing we have. So what they would look for is a healthy member of the same household, and that person would have similar gut microbiota, but they're healthy, so their microbiota is not making them sick. They have to test them to make sure they don't have gut viruses and dangerous gut bacteria or fungi. Like maybe the donor's immune system is keeping these things under control, but the recipient doesn't have immunity to them. That's always a possibility. But if it seems that the donor is safe in that way, they will give antibiotics to the recipient and clear the dysfunctional community from them, surgically transplant the new community, or that would be through an enema or something similar, and then cross their fingers, hope it works. And that's kind of where we're at with controlling our microbiota. We have extensive, huge amounts of research into it. In the 90s, we thought we would understand it very well. We got these new technologies that could describe what bacteria are there and what they're doing. But every new technology we get shows us that the microbiota of the gut is more complicated than we thought it was. It's on the scale of the complexity of a brain, except that it does more functions than a brain does. So those are the two things that we would really like to understand fully, but we can't. In our lifetimes, we're never going to be able to model a human brain fully, and we're never going to be able to fully understand the microbiota or how to control it. So what does the research look like? Well, we have experiments in mice and rats, and these have been amazing. They've found really cool, compelling effects microbes can have in the guts. So feeding specific microbes to mice can give them or remove an autism-like syndrome. Bacteria you can feed to mice will give them plaques in their brains, in their nervous system tissue, that are similar to the plaques that develop during Alzheimer's. There are, one of the earliest studies showed that by feeding different bacteria to mice, you could give mice extreme obesity, or make them lose the weight by giving them different bacteria. So imagine, you read these studies, you immediately want to see, okay, can I eat something different that will help me gain lots of weight or lose lots of weight? Can I eat bacteria that will save me from Alzheimer's? Or cure my autism? And the answer right now is absolutely not. We have no idea how to control things like this, and we don't know that these effects would happen in humans. We don't have the great evidence that there's something we can do in humans that will have an effect like what happens in mice. So we get these compelling effects in mice, but they don't translate, or generally haven't translated to humans. What we have in humans are a lot of observational or correlative studies where we describe the bacteria in guts and correlate them to whatever health data we can get from the same subjects. And so that's how we got the list, like this list. These are correlations between bacteria we find in the gut and other health. So ultimately, between our very complicated immune systems and the complicated diet we eat and the complicated bacterial communities we have, they are doing things we can't understand, and anything we do to change those communities has effects that we can't predict. So I thought, when I went to graduate school, I thought I was part of a new revolution that would break this apart and understand it fully. And what we now know is that is not happening because it's just too complicated. And so instead, we're looking for things like fecal microbiota transplants, or doing experiments on individual probiotic strains and things like that to see what we can eat that will improve our health. So that leads us to prebiotics, probiotics, and fiber. There's a big industry around selling probiotics. These are living, beneficial bacteria we would eat. And my analogy for this is throwing seeds into a forest. Somehow, there are some bacteria that when we eat them, they improve our digestive system. So maybe less gas, maybe less constipation. And there are some experiments, basically, these are not regulated as drugs, so we can't have the same medical studies, and they can't have the same strict conclusions that drug companies can get. They can't be prescribed as treatments for diseases, though they are sometimes. We don't really know how they affect our health. We don't know what they do, and most of them just die in the gut. If you throw seeds into a forest, what's going to happen? Squirrels will eat them. Maybe one or two will successfully turn into a new plant. Yet somehow, there is a lot of evidence that probiotics can help with our digestion. Claims like they can help relieve anxiety, I don't know. There are experiments where specific probiotic strains have decreased anxiety and other mood-related things. So the problem is all those experiments are done by the companies that sell the products. So I don't know, take it with a grain of salt. A prebiotic is something that you eat that we don't absorb or digest. So it's not a living thing. It's like dietary fiber, like carbohydrates. They pass through the gut, and our beneficial microbiota can eat them and use them as a specific energy source. So this is giving our beneficial microbiota an advantage they wouldn't otherwise have over their competitors. And so I think of this as like increasing the sunlight shining on a forest, or increasing the amount of rain that falls in a forest. And somehow they have a good effect. The tricky thing with this is we don't 100% know which microbes are beneficial. So we don't have a lot of great evidence that prebiotics are good for us. We have enough evidence that I eat prebiotics. I mix inulin and wheat dextrin into my cereal in the morning. And occasionally I make fermented foods. I will make kimchi and I eat sauerkraut. I wouldn't bet my life that those things are good for me at this point. But they haven't killed me yet, so there's that. And then dietary fiber is a really complicated topic. Several years ago, I worked at a private college, and I carpooled with professors, and they all wanted to know what we should eat to change our health. And so I read a lot about fiber and prebiotics, and what I can tell you is it's really complicated, but lots of types of fiber we can eat will help with gut health. Lots of them will prevent constipation or limit the amount of gas you have. And so if you limit yourself to trying to improve your digestion, probiotics, prebiotics, and fiber can do that. You can improve your digestion. Anything beyond that, I think it's premature to assume we can do that by eating things. Okay, so that's it for the resident microbiota. I know it's a big topic and I talked for a long time. I wish I had more things I could tell you to eat that would be good for you, but it's not responsible of me to tell you those things because the data don't seem to be there. But the mouse studies are just incredible. The things we can do to mice by feeding them bacteria are unbelievable. So cross your fingers and pray that some of those things can translate to humans. That would be amazing. I'll see you at the next video.